Come on in, ladies. All right, guys. Computer's a little slow this morning. It'll catch up in a minute. Two topics today, mostly. A little bit about where cells supposedly came from according to the current theory. And then some biochemistry stuff. That's what we're going to concentrate on today. So this thing will catch up with us in a minute. Well, thought it would. Here we go. Get that out of the way. Don't know what that is. All right, so scientists think that in the beginning there was just like water, right? And inorganic molecules. What's that idea that things like lightning and gases all got together to make amino acids, they got together to make proteins, got together to make cells? What's that idea called? Yeah, it's called operant hypothesis. So let's look at see what we can do. So here's the idea that in the beginning you've got these gases in the atmosphere. You've got lots of lightning and heat and stuff from like volcanic activity. Pretty much no oxygen in the beginning. Somehow these molecules reacted together to make monomers. Remember, monomers are things like what? What are some examples of monomers? Amino acids. Keep going. What kind of carbohydrates? Monomers. Monosaccharides. What about for like DNA? What are the monomers there? Nucleotides. So these chemicals supposedly got together to make those really simple organic molecules. And eventually, those came together, the monomers, to make polymers. Things like proteins and DNA. And then somehow those came together to make the first cells, which according to this idea would have been what kind of cells? Bacteria, prokaryotic. Also remember that the idea is that the first genetic information was what kind of genetic information? RNA. And there aren't any living things on Earth today that are based solely on RNA. Because what's the problem with RNA? It mutates a lot. It's not stable like DNA. And most of the time mutations are bad, right? Occasionally you'll get a good one. But most of the time mutations are a bad thing. So over time, once DNA developed, things evolved to be DNA based because it's more stable and more dependable and less prone to having mutations. All right, so Operin is the guy who essentially came up with this idea. His name's Alexander Operin, came up with it in 1924. There's another guy, John Haldane, that was in on it too, working together. But essentially they said that the original atmosphere of Earth didn't have any oxygen in it. It had stuff like ammonia, carbon dioxide, and that with the help of lightning and ultraviolet light and maybe heat from the Earth, you got organic molecules, amino acids, nucleotides. Those came together to make more and more complicated, bigger macromolecules until you eventually got cells. Now, there's a famous experiment that they've asked about several times in the AP exam that tried to test this hypothesis, and it's the Miller-Urey experiment. And what Miller and Urey did is they took the gases that Operin and other scientists at the time thought were in the initial atmosphere of Earth. Things like methane. What's the formula for methane? CH4, ammonia, NH3, water, and some hydrogen gas, some H2. They put it in a closed gas container. They then put in essentially a spark plug up top, hooked up to a battery. That was supposed to simulate what? Lightning. Let it sit for quite a while. This is continuously sparking. And when it was all over, they looked at what they called the organic soup down here, and they were able to find what kind of molecules? They found some amino acids and even some nucleotides. But no way. No cells, no proteins. But at least they backed up part of the hypothesis. That was their idea. There's a lot of debate about this experiment because now most scientists have decided that these probably weren't the gases in the early atmosphere, which means, does this experiment have much credence? Not really. That's the Miller-Urey experiment. Its job was to try to back up some of what Oparin had come up with in his hypothesis. Also make sure you know, we think prokaryotic cells came first, and we think the eukaryotic ones came from them. And there's two ways to explain how that happened. At least these two probably work together. The one we spent the most time on is the endosymbiotic hypothesis. And that's essentially the idea that big prokaryotes did what? Yeah, took in smaller ones, phagocytized them, but didn't digest them. And that over time, those two things started working together. The little ones inside the big ones. And turned out to be a good deal for both, a mutualistic kind of relationship. And the little ones eventually became what? The organelles inside the big ones. And that's where eukaryotic cells came from, at least partly. There's lots of pieces of evidence to try to back that up. There are today bacteria that live in symbiotic relationships with other bacteria. We know that. It's provable. We know that mitochondria and chloroplast have their own DNA, separate from that in the nucleus, and it's circular, like bacterial DNA. We know that mitochondria and chloroplast can reproduce independently of the cell they're living in, almost like binary fission, like bacteria. And also, that mitochondria and chloroplast have ribosomes in them, separate from those in the rest of the cell, that seem to be more like bacteria ribosomes than eukaryotic ribosomes. There's also this idea of membrane invagination. This thing's really not cooperating today. Oops. Endosymbiosis doesn't explain all the organelles. It explains some of the ones like maybe mitochondria and chloroplast, maybe even flagella. But some of the organelles that are essentially just like storage containers, it doesn't really explain those. The idea of membrane invagination is that the cell membrane pinched inward on itself and essentially formed little baggies, right? Little containers. And that those became organelles. That explains some of the organelles like vacuoles, like the Ruffyar, the lysosomes, for example, all could have been formed by membrane invagination. So the big idea is that these two processes worked together to make modern eukaryotic cells, endosymbiosis and membrane invagination. We're going to switch gears a little bit now and talk about some biochem stuff. So first of all, pretty much all living things are based on what element? Carbon. Now there's 100 and something elements on Earth, but every living thing that we know of is based on carbon. So there's got to be something unique about carbon that makes it important for building living things. So one thing you ought to explain is why carbon. You ought to be able to explain that. There's several things about carbon that make it good. One is there's a lot of carbon on Earth. It's abundant. It's not the most abundant thing on Earth, but there's a lot of it. Another important thing about carbon is it's a really small atom. It's a non-metal. What kind of bonds is it mostly going to make? Covalent. And since it's small, what does that mean about those bonds? They form close to what? Close to the nucleus, which means they're held on to really, really tightly, so they're strong. If you were designing a building, you want to build it out of something strong. And that's sort of what carbon is. It's a strong building material. Another thing about it is it's in column 4 in the periodic table, column 4A, which means it's got 4 valence electrons. What do we know happens with valence electrons during covalent bonding? They get shared. What that means about carbon is it can bind to at least what? 4 different things at one time. And then what that allows it to do is make molecules in lots of shapes. And one of the things we talked about this whole class is that shape determines function. So if it can make a lot of shapes, it can make a lot of molecules with different functions. So that's pretty important. All those things are partly the reason that it's carbon that we're all built out of. All right, something else we want to talk about. Most organic molecules are these big molecules. What are the words for those? Macromolecules or polymers. And they're built usually out of these repeating parts we call monomers. There are reactions that occur in pretty much every living thing that put together monomers to build polymers. And we can see that going on right here. So what happens in these reactions is this OH from one of the monomers and the H from the other, those are removed. That frees up a bonding area, a bonding site. And then those 2 monomers get stuck together. Water is released. We call these kinds of reactions that put together things condensation because they produce water. Sometimes they're called synthesis. What's the other word? Dehydration. All of those are reactions that put together monomers to build polymers. We also might call them anabolic, right? Because anabolic means to what? Build up. The opposite of those reactions are the ones we see over here. Hydrolysis reactions use water to break things down. So they take a polymer and they break it down into its monomers. We got to have water as a reactant to break those bonds. What are these typically going to do with energy, the hydrolysis reactions? Are they going to take it in? You're breaking bonds. Usually what are you doing with energy? Releasing it. So these are usually exergonic reactions, whereas these are usually endergonic. Because usually you have to add energy to make bonds, right? What kind of delta G value would these usually have? The hydrolysis reactions. Negative. And these usually going to have a positive. Does that make sense? All right, going a little further. All right, not going to talk too much about carbohydrates, but just quickly a little bit about them. In the body, carbohydrates are sugars. They're used for quick energy supply and also to build things. The most simple carbohydrates are monosaccharides. And what's the most famous monosaccharide? Glucose. But there's also ribose and deoxyribose and fructose, and there's all kinds of different monosaccharides. But they're the building blocks. Here we see glucose on the left, fructose on the right. These are isomers of each other. Because what's the same for both of them? Their formula is the same. They're both C6H12O6. But what's different is their shape, and therefore also their function. Here we see condensation going from left to right. So we're putting together 2 glucose molecules to make a maltose. And then from right to left, we've got hydrolysis. We're breaking that disaccharide down. We also want to talk about polysaccharides. And there's really 3 of those you ought to be familiar with. One of them is starch. Starch usually comes from where? Where in nature do we find starch? Plants. And plants are using starch as a form of what? What do they make the starch for? Yeah, I heard two things. I heard storage and structure. Which one's right? Starch is about storing energy. Think of a potato. It's mostly pure starch. It's not about structure. It's about storing energy for baby potatoes. Corn, same kind of thing. Rice, same kind of thing. Starch is about storing energy. And each one of those little hexagons is a way. So each little green hexagon up there.
Teachers and students, if you have parked in front of the gold gym, the first two rows, Coach Gurley needs you to move your vehicle in order for the blood drive vehicles to pull in. Thank you.
All right, another one that you need to know about, another polysaccharide is glycogen. It's very much like starch. It's also used for what? Storage. But it's used for storage where? Humans and animals in general. And where do we tend to store a lot of glycogen at? In our liver and also in our muscles. And we've got a couple of hormones that tell us when to make it and when to break it down. What's the main hormone that tells us we need to be making glycogen? Insulin. Tells us to take glucose out of the blood and make glycogen out of it, right? Store it. And then there's two hormones we talked about that tell us when to break it down. What are those two? What's the opposite of insulin? Glucagon. That's just normal every day. And then what's the one that tells us we need to break it down, especially in times of stress? Epinephrine or adrenaline. Another one that you ought to know about is cellulose. Cellulose is the one that's about what? Structure. Mostly found in plants, specifically in the cell walls. It's made out of glucose, but the glucoses are put together in a different way. They're put together in a way that you not only have covalent bonds going horizontally, what kind of bonds you got going vertically here? Hydrogen bonds. So this makes this much, much harder to break. It also makes it much stronger. This is not about energy storage at all. What do we know happens when we eat cellulose? We can't digest it, it just comes out. It's fiber. This is not about energy storage, it's about building things. All right, we talked a little bit about lipids yesterday, so we're not going to say too much about them today, other than the fact that lipids are this big group. They don't have a whole lot in common. The one thing that they mostly have in common is that they don't dissolve in water. They don't dissolve in water because they're mostly what? Non-polar. Lipids don't like water. Water's polar, lipids are not. They don't mix. Lipids include fats, oils, wax, phospholipids, and steroids. We're going to focus on just maybe two of those right now. But their main function is to store energy for the long term. It's not about short-term energy storage, long-term energy storage. Most important one probably is just a regular old fat. And this is what a fat might look like. It's what we call a triglyceride. It's tri because there's three what in there? They're called fatty acids. Those are the yellow parts. They're just carbon hydrogen chains. And they're attached up to this thing called glycerol, which is in green. And that molecule is really, really good at storing energy, mainly because it has a whole lot of what? Binds. And in chemicals, that's where energy gets stored, is in binds. And to get the energy out, what do you got to do? Break them. You got to break the binds to get the energy out. All right, we also talked about phospholipids. We know these are found in our membranes. And what's the word that is associated with phospholipids that we ought to know? Amphipathic. Because one side of them is what? Polar, and the other side isn't. So when we put them in water, we know they form a bilayer, phospholipid bilayer like that. And that same structure can be found in pretty much every living cell. There's steroids. three steroids you ought to be familiar with: cholesterol, testosterone, and estrogen. All the steroids are built from cholesterol. This is the starting molecule. And usually, their main function in the body is as hormones, sex hormones. But cholesterol also has a role in your cell membranes. It makes it a little bit thicker than what it would normally be. We're to focus mostly on proteins, because that's the one they like to ask the most about on EP exams. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and sometimes other things, like what? Sulfur especially. If I ask you for a use of proteins in your body, what would be a good answer? Pretty much everything you do happens because of proteins. Pretty much every part of your body is made of proteins. But the most important ones are enzymes. Enzymes are proteins. Proteins are lots of proteins, at least are enzymes. What's a word we ought to always associate with enzymes in terms of their functions? Well, activation, energy, true. That's not where I was going now. Because of what they do. What's a one-word descriptor of enzymes? Catalyst. And that means they do what? What does it mean to be a catalyst? It means you get things started, right? You get them started. And they get them started by lowering what? Activation energy. Remember that our proteins are built out of amino acids. And in nature, there are 20 of those. 20 naturally occurring amino acids that you either have to get in your diet or you have to be able to make yourself. And that's true for every living thing. Every living thing is made out of these 20. Let's look at a picture. You need to be able to draw an amino acid if you don't already know how to do that. Here's what they look like. Here's our amino group. What's the real name for that group right there? Carboxyl group. There's a hydrogen atom, carbon in the middle, and then this R group is the part that's different between all 20 amino acids. It's the part that makes the 20 different from each other. The part I'm circling here is exactly the same in all 20. This part is different. Now sometimes you'll see it drawn when it's neutral, like this. Sometimes you'll see it drawn when it's ionized, like this. Either way, these are amino acids. All that's different between these two pictures is here the carboxyl group lost a hydrogen, so it acted like a what? Since it lost a hydrogen. Acid. And the amino group gained a hydrogen, so it acted like a base. So amino acids are actually acids and bases at the same time. What's the word for things like that? It can be acidic and basic. Amphoteric. So, that's just showing you the different R groups and what they might look like. One important thing to remember, and one thing that determines the shape of a protein. Let's say this is a chain of amino acids. What's the word for that? A chain of amino acids. A polypeptide. And let's say that this middle blue amino acid here in my chain is one of these amino acids that are hydrophobic. Has a hydrophobic R group, which means it's not polar, right? When this thing goes into the rough ER to take on its shape, where does that blue one need to be? It needs to be folded up in the middle, away from what? Away from water. And that's one of those things that determines the shape of a protein, the polarity of the R groups of the amino acids. Let's say there was a mutation, though, in the DNA that coded for this polypeptide chain. And instead of getting one of these amino acids here where this blue one is, we got one of these, the hydrophilic ones. That's going to completely change what? The whole shape. Which means, is this protein going to work anymore? Probably not. So one of the reasons that proteins fold the way they fold is the polarity of their R groups. If that changes, even just one of them changes in a chain, that can completely change the whole shape of the protein. Remember that proteins get folded either in the rough ER or, there's some proteins that are made just in ribosomes that are floating out in the cytoplasm, right? They have these little tube-like structures called chaperonins that they go into and get folded. Either way, the way they get folded is hugely important. And we've talked several times about the different levels of structure in a protein. Let's go back through this again. The most basic level of structure is primary. This is the polypeptide. What makes up its primary structure? Yeah. Which amino acids are in there? Like what order are they in? Which amino acids are in this thing? What's the order of those amino acids? That's primary structure. That is the only level of this that's determined solely by what? DNA. That's what DNA codes for. Primary structure. Secondary structure is how are certain pieces of this chain shaped. Some parts of this chain would really be really long. Might be shaped in a helix. Other parts might be like mountains and valleys kinds of things, like a sheet, a pleated sheet. But the secondary structure is how are certain parts of this chain shaped. And that shape, that secondary shape, is mostly held in place by hydrogen bonds. Because what do we know about amino acids? They're what kind of chemicals? What kind of chemicals can make hydrogen bonds? Polar. They're polar. But again, what do we know about hydrogen bonds? They're not all that strong, so this can be changed. The tertiary structure, the third level of structure, is the shape of a single polypeptide chain, the overall shape of the whole chain. Not of parts of the chain, but the shape of the whole chain. And there's several things that can determine that. Let me get to a picture. Again, this tertiary structure is held in place by hydrogen bonds, but also these little orange lines, they represent what? Disulfide bonds. Remember that some of the amino acids have sulfur in them, in their R groups. And that if two of those get near each other in a chain, they connect, and they form a strong bond, harder to break than hydrogen bonds. So it's like almost like scaffolding, holds this shape in place. Tertiary structure, the shape of one polypeptide chain. But typically that's not a protein, because most proteins are made of several of these polypeptide chains that are all wrapped together. And how the different chains wrap together, that's what we call the quaternary structure. And we can see that there. So we got two chains here. We got the dark blue and the light blue. How those different chains have wrapped together make up the quaternary structure. What do we call it when something causes that shape to change? Any level of this shape to change, what do we call it? Denaturation, right? And what are some things that can make a protein denature? Some environmental things that can make it denature? Heat, pH, what'd you say? What else? Heat, pH, salinity, saltiness. Any of those can make it happen. And usually, which levels of the structure are changed in denaturation? What are the two that would be easiest to change would be the two that are dependent on what kind of bonds? Hydrogen bonds. And which two levels are dependent on hydrogen bonds? Secondary and tertiary. Those are the ones that usually get changed in denaturation. And we know if a protein gets denatured, it's not going to work anymore, right? All right, so we'll mention a little bit about nucleic acids, and then we'll call it a day for today. Nucleic acids are our fourth category. There's several different ones of these, but they mainly have two jobs. They're either about storing and passing on genetic information, like DNA and RNA, or they're about transferring and storing energy, like ATP, NADH, FADH2. Those are all examples of nucleic acids. The monomers in these guys are nucleotides. Those are the building blocks. And several times during the course of this class, we've looked at this same exact picture of a nucleotide. And hopefully you know that in a nucleotide, in the middle of it, there's a sugar. And it's usually a pentose sugar. Pentose meaning what? 5 carbons. They're numbered, usually clockwise. Attached up to carbon number 1 is always one of these guys, a nitrogen base. In DNA, we know those might be A, T, C, or G. In RNA, they might be either A, U, C, or G. If this was ATP, what's the nitrogenous base? Adenine. It's going to be A. And then attached over here to carbon number 5 is always a phosphate, which makes the nucleotide be very what? Negative and very polar, both negative and polar. And those turn out to be really important in terms of how they work. So with DNA, I'm not going to say too much about DNA because we spent a lot of time on DNA. But we know DNA's only real job is to code for proteins. You want to think of DNA like the blueprints for you. We know that it's stored in the nucleus, except for what part? What part of your DNA is not in the nucleus? The mitochondrial DNA, which you got from where? From your mom. Only from your mom, not from your dad. We know that DNA's got that double helix shape. And what connects the 2 strands of DNA together? Hydrogen bonds between the bases, A to T and G to C. So here we can see sugar, phosphate, and nitrogenous base. That's a nucleotide. Here's another nucleotide. Sugar, phosphate, sugar, nitrogenous base, phosphate. You got 2 chains. This chain over here connected up by hydrogen bonds to the chain on the left. RNA looks much the same, except it's only what? 1 strand instead of 2. And there's lots of different types of RNA. We really only talk in much detail about 3 of them. We know that the messenger RNA is a messenger between what 2 things mainly? Messenger RNAs carrying messages between nucleus and ribosomes. The tRNA's job, the T actually stands for what? Transfer. But I told you to think of it like what? Taxi. Because what's the job of tRNA? Carry amino acids and put them where they're supposed to be, right? Deliver them to where they're supposed to be. What are the 3-letter code words that are found on a tRNA called? Anticodons on a tRNA. And each 3-letter code word on an RNA, mRNA, is called a codon, right? 1 codon codes for 1 what? Amino acid. All right, let's talk a minute here about ATP. Here's ATP. We know it's also a nucleic acid, but it's different than DNA and RNA because it's just made of 1 nucleotide. It's a monomer. It's not a polymer. We can see in this picture, here's adenine right here. This is ribose, same sugar that's in RNA. It's got 3 phosphates. And then going from in this direction, what kind of reaction is happening to ATP? Hydrolysis. So breaking it, which means we ought to be giving off energy. what's the big category of enzymes that makes this reaction go left to right? Protein kinases, right? They take away a phosphate, and they'll add that phosphate to something else and activate it. It's a good way to transfer energy, free energy. Going this way, we're making ATP. We're taking ADP and putting it together with P to make ATP. What's the word for that kind of reaction? Going right to left. We could call it a dehydration, condensation, but what's a more ATP-like word? Phosphorylating, right? We're phosphorylating. And in photosynthesis and cell respiration, what's the main process called that makes the ATP? It's the process that uses ATP synthase and hydrogen ions. What's that process called? Part of the electron transport chain, but more specific to ATP, which is chemiosmosis is what I'm looking for. Chemiosmosis. It's the main process in your body for making ATP. You get energy from those moving protons, moving H pluses. All right, we did that already. Let's go back. The word here on the bottom of the screen, central dogma. Let's just remind ourselves of what that is, and then we'll let you go for today. The central dogma is like the core idea of how genetics work. And as far as we know, every living thing on Earth has DNA. But that DNA is stuck where? In the nucleus, at least in eukaryotes. The DNA, first of all, has to be used as a template for making the RNA, the mRNA. And we call that process transcription. After transcription happens, at least in eukaryotes, what has to happen to that mRNA before anything else can happen? It's got to be processed, which means what has to be taken out? Intron sequences. And what do we call the enzymes that do that? Spliceosomes or ribozymes or SNRs. There's like 3 different categories of them. Once that's processed, though, and ready to go, the mRNA is going to be used to make the proteins. And that process is called translation or protein synthesis. And it happens where? At the ribosomes. And you know, those proteins go on to determine pretty much everything about you. They determine your traits. And then we'll throw in this little loop here to represent what? Replication. And we know when during the cell cycle does replication take place. What part of the cell cycle? What part of the interphase? S phase is interphase, right? And that's where we'll stop.